Good day, dear students. We are at present studying about the antigens and antibodies. In the previous lecture, I discussed regarding antigens, their definition, different types of antigens, and how these antigens behave when they enter into the host cells. You must be remembering that in the lecture, I gave you the definition of the word antigen as well as immunogen. I think the more proper word would be immunogen, but loosely it is many times spoken as antigens. And how was it defined? It was defined as the substance, normally foreign substance, which when enters the host will respond by producing a corresponding and specific immune response. And these responses can be either by T cells or B cells. The only difference between the two words, antigen and immunogen, was immunogen elicits or provokes the immune response and will react with the antibodies so produced. While antigen, normally we restrict these words specifically for those kind of substances which may or may not be able to elicit the immune response, but it has the ability to react with it. So the point is that whenever a foreign substance tries to enter into our body, that is in the host's body, our means I'm talking about the host as a human being. Human beings, they do have many ways to foil the attack, to stop that. And we had discussed earlier various first line, second line of defense mechanisms like skin, mucous membranes, hair, various enzymes, normal flora, phagocytosis, etc., etc. The third line of defense, or a very specific defense process, was the antibodies. So, I am going to discuss in detail antibodies, the structure and the functions. Now, as it is shown in this title, you must be seeing various two-armed Y-shaped structure. Normally, Y-shaped structure which is there is also attached with another small arm. So, this Y-shaped structure, normally in diagram, it is depicted in immunology as a typical structure of antibody. And that is why the title represents what we are going to study, that is antibody, its structure and functions. Antibodies are also popularly known as immunoglobulins. In short, it is written as IG, I capital and G small. Immunoglobulins. Globulins means it is a type of a protein, and immuno means the one which is responsible for imparting some immune response. And therefore, this word is derived from those two separate words, immunoglobulin, written as IG. Now, what are those immunoglobulins? So, immunoglobulins, you can define it as glycoprotein molecules which are produced by plasma cells in response to immunogen and is responsible for immunity. Now these are the key words in the definition. First one, they are glycoprotein in nature. The chemical nature is glycoprotein. It means some sugars plus proteins, glycoproteins. And they are produced by which cells? Plasma cells. Plasma cells are the type of B cells. B cells will differentiate into plasma cells and which is responsible for producing such glycoproteins. And when are they produced? They are produced in response to immunogen. It means whenever an immunogen comes across in the body, there are defensive cells which counters it by production of such molecules. And what is going to be its main function? Its function is to impart immunity. It means to impart resistance. How it is going to impart the resistance? Because it is very simple that these glycoproteins which are produced in response to the immunogen will bind with them, and as a result these immunogens, that is the antigens or the foreign molecules, are destroyed by different mechanisms. So again, if I define it, it is a glycoprotein molecule which are produced by plasma cells in response to immunogen and is responsible for immunity. So this is a definition of immunoglobulin. And it constitutes almost 20% of total proteins in a healthy individual. And therefore, you can say it is not a small amount. It is a substantial chunk, that is 20% of the total proteins in a normal individual. It is consisting of immunoglobulins. Now, let us discuss in brief how these immunoglobulins were searched or discovered. This was done by the experiment performed by Tiselius and Cabot in 1949. This was one of the classical experiments in which the electrophoresis of a serum from an immunized animal was performed. When a serum you know that it is something which is obtained from blood. When the blood is collected, is left undisturbed, the upper portion, that is fluid, cellular materials will sediment at the bottom, and the fluid like portion, which is slightly pale yellow in color, is a serum. It's a fluid material in which these formed elements, RBCs, WBCs and platelets, they circulate in our capillaries. So this serum, when it was electrophorized, that is electrophoresis was done, then 4 different fractions were obtained. Which were those 4 fractions? 1, albumin; 2nd, alpha globulins; 2nd, beta globulins; and the 3rd, gamma globulins. So 3 portions of alpha, beta and gamma globulins, and 1 was albumin. All of them, they are proteins. Albumin is also protein, and this alpha globulin, beta and gamma globulins are also proteins. So when you obtain such 4 peaks during electrophoresis, it means these 4 proteins were separated by electrophoresis. Then What they did further was the albumin, oval albumin, it was removed by precipitation, and then antiserum was electrophorized again. So point is, they removed albumin fraction by precipitation and then again electrophoresis of that things. These gamma globulins were the three components, alpha, beta and gamma, out of which the slowest one, that is the one which moves very slowly. Which one will move slowly? One which has got a least charge. So this slowest moving group of proteins, and it were present at a much lower concentration after precipitation. And why they were named as a immunoglobulins? Because it was found that these proteins which migrates, they were the proteins, they imparted immunity. As you can see in this figure, this is a graph which is depicted, what we call it as a graph from electrophoresis of serum. So on the y-axis we have amount of protein, and this is the mobility on the x-axis. So the highest amount was of albumin. Once it was separated by precipitation, what remained were the fractions of globulins, and in that fractions, this one, that is the alpha, it is further categorized as alpha 1, alpha 2, this one. Then we have beta fraction, and then this is the one which is gamma fractions. Now this fraction was the one which was imparting immunity, and therefore it was known as immunoglobulin. This is the one diagram which is shown. Now with this brief introduction, how they came into existence or how it was discovered, mainly it is to honor two scientists, Theselius and Cabot, and this experiment was done almost 70 years ago, 80 years ago. Now let us come to the structure of immunoglobulins. Now if we see the basic immunoglobulin structure, I am using the word basic because as we will see in detail, there are different types of immunoglobulin which varies in structure from one to another. But in all these different types, they have one basic structure. And therefore, let us study the basic structure of immunoglobulin. The first thing that one should say that it is made up of 4 peptide chains. You know peptide is a short chain of amino acid. Short means few hundreds amino acids joined together in a linear mode, in a linear fashion by CONH, that is a peptide linkage. So 4 peptide chains are there. Out of these 4 peptide chain, there are 2 identical chains and they are known as light chains. L. Light chain represented as L, and their molecular weight is approximately 22,000 Daltons. Second one is 2 heavy chains whose molecular weight was almost double that of light chain, around 55,000 Daltons. Now, because the molecular weight was very high, naturally it was labeled as heavy chain. And because other two chains were having a low molecular weight, they were called light chain. So out of 4 peptides, 2 were heavy and 2 were light. Okay. Now, are these chains separate from each other? No, in no way. These light chains, they are bound to heavy chains by disulfide bond. What is that? It is a non-covalent interaction as salt linkages. Apart from disulfide bonds, hydrogen bonds and hydrophobic bonds will bind light and heavy chain together, and it will form a heterodimer. Dimer. Dimer means 2 chains together, and hetero means these 2 chains are different. One is light and the other one is heavy. So this heterodimer part is known as HL heterodimer. I hope this is clear. So there are 2 light chains and 2 heavy chains. Light chains were bound to heavy chains, and that becomes HL dimer, heterodimer. Similarly, another light chain will bind to a heavy chain. It will become another heterodimer. Now, these 2 light and heavy chains are again binding with each other. That is, heterodimers, they also bind with each other. So, similar non-covalent interactions and disulfide bridges link 2 identical heavy and light chain, that is HL chains, together, and they will form the structure of basic 4 dimers, that is HL2. Now, this we call it, suppose that it is represented here in this diagram very clearly. This is a light chain because of its small size, while this one is a heavy chain. These light and heavy chains, they are bound with such kind of disulfide linkages. So this as a whole becomes what we call, this as a whole becomes one heterodimer, HL dimer. This is the one. And similarly, exactly in juxtaposition opposite to it, you have this light chain and this heavy chain. As a result, as you can see, so as we are seeing in this diagram, this is one HL dimer, this is another HL dimer, and both of them, they are linked with one another by again certain disulfide linkages, which are as shown here. One, this is the one, this is the another one. So basically, this is bound here, this is bound here, and these heterodimers, they are also binding with one another by disulfide bridges. So this is how 4 different peptides, I'm using the word peptides, they exist together to form one molecule, and this is the basic structure of immunoglobulin. Okay, we will further see its detail. In this diagram, as we see that the light chain is having 2 parts. We divide it into 2 parts to make the system clear. Actually, it is one peptide chain. But for our understanding, we can label them into two parts. This is known as variable region, VL, variable light, and this is known as constant region. As you can see in this diagram, the one which is having a yellow color is a variable region, and the one which is having a red color is a constant region. Now, as the name indicates, what do you mean by variable? Variable means it must be changing. So, the amino acids located in this region, they change depending upon the type of immunoglobulin, depending upon the immunoglobulin produced by different immunogens. So that is why it is known as variable. Similarly, in a heavy chain, this one which is a heavy chain, this as a whole is also divided into 4 sections. The one at the top is known as variable heavy chain, followed by 3 sections which will be constant. So constant heavy 1, constant heavy 2, constant heavy 3. In this way, the heavy chain has 4 sections for our understanding, and light chain have 2 sections. The one at the distal end, where actually it will bind with an immunogen, this region as a whole is known as variable region, because this variable region is allotted to light chain also and heavy chain. And again, I am reminding you that variable means the amino acid, sequence of amino acid in this region, it may alter. They are not same. While in case of constant region, the sequence of amino acid in this peptide more or less remains same. There is a very little changes, and this is responsible for what we will call later as diversity of antibodies, because any foreign material can act as an immunogen provided it fulfills certain criteria that we discussed earlier, like size, that is the molecular weight, foreignness, its rigidity, structures, ability of digestion, all those things. So there are innumerable immunogens, and naturally the host should have ability to produce similar innumerable huge amount of immunoglobulins. Then and then they will be able to bind with one. So this is the variable region which plays an important role in what we call it as a antibody diversity. Okay. Now, these two arms, which consists of variable light, variable heavy, constant light and constant heavy 1, these are plus these two, it forms the complete structure. Now, if we go further in detail, then you will find that this portion, as I have told you, variable region, this is the one variable region. So this variable region is also called as antigen binding site. I just told you it is responsible for binding with the immunogen or antigen. While this portion, which one? The one which I am showing you here, this one, is responsible for its biological activity. Means when this antigen binding region, when it searches and binds with the specific immunogen, then there will be certain signals sent to this biological activity, and it will give certain reactions, biological activity. This region, as it is known as one which is responsible for biological activity, they are also known as FC region, fragment constant. And this is the fragment variable. So this is variable, and this portion will as a whole be called as a fragment constant, FC region, FC. Okay. So variable region is at the top of the arms, and normally it is approximately 110 to 120 amino acids long at the N-terminal region. We will have to define top means what. I just told you at the top of the arm, but in scientific language we can say that at N-terminal region, NH3, N means NH3, because when there is a peptide, there are two ends, one is N-terminal and the other one is C-terminal, because peptides, as I have told you, they are made up of amino acids, and amino acids bind with one another. One amino acid binds with the another by peptide linkage, and this linkage is between carboxyl group and amino group. So at one end there will be amino group, and at the other end, one end will have free amino group and the other end will have free carboxyl group. So we call it as a N-terminal and C-terminal. So at N-terminal, these variable regions are located. And what is their length? The length is approximately 110 amino acids. Constant regions, they have, as I have told you, no diversity. The sequence of amino acid is more or less same. I am using the word more or less. That does not mean that it is exactly same in all. There is a little bit of diversity, little bit of variation in sequence of amino acids. And again, it is in case of light chain, what is the size? Approximately 110 amino acids. While in case of heavy, it was, because there are three: constant heavy 1, constant heavy 2, constant heavy 3, three regions. So it is approximately thrice the light chain. So it is 330 to 440 amino acids. Now, another important thing is the presence of a hinge region. What is hinge? Hinge is something, if I give you example, let us consider door or a window. The door is attached to a wall on a frame, and in a frame there are 3 to 4 hinges through which this door is attached to that frame. It can be door or it can be a window. Now, and that is the reason that we are able to close the window, open the window, or we can partially close the window of our liking. More, less, slightly more, slightly less, or totally closed. So it is the hinge which will, hinge means a kind of a joint. So this is a hinge region. If it is completely closed, it will be like this. If it is open, then this will be this. Or it can be partially closed, partially open. So that is the hinge region. And this hinge region, what is the function? Naturally, their basic function is to provide flexibility in the molecule. Now, this hinge region is proline rich region. Proline, it is a kind of an amino acid. And mu and epsilon heavy chains lack the hinge region. What are mu and epsilon heavy chains? These are the different types of heavy chains which has been labeled as mu and epsilon. Like light chain, as we will see, they are also labeled as kappa and lambda. But don't go into that details for your understanding. Simply understand that there is a hinge region. So this is a hinge region. In diagram, I can show you that these are the hinges. And therefore, through that hinges, these molecules, that is the antigen binding sites, depending upon the size of antigen, it can open up or slightly close. Angles can vary. So this is a hinge region. And this black colored one, it is proline rich region. Amino acid, which one? Proline. Okay. Now, I told you in the definition that immunoglobulins are glycoproteins. Then you may ask a question that so far what we have discussed are the 4 peptide chain. Where is the sugar? Now I will tell you that there are certain sugar molecules attached to CH2 domains, means constant heavy 2 region. Where are these? At constant heavy 2, that is here, this one. At these junctures there are certain sugar molecules, as you can see in this figure. See, CHO. CHO group means sugar groups. This is CHOO, that is the C terminal. This is N terminal. So whenever an antibody structure is asked in exams, you should exactly remember this diagram. It shows everything. N terminal, C terminal, light chain, heavy chain, light variables, heavy variable, constant regions, constant light, constant heavy, constant light, then constant heavy 1, constant heavy 2, constant heavy 3. So remember this diagram properly. This is the same thing which is shown in with details. 2 heavy chains, 2 light chains. This is light chain, this is heavy chain, and basically this forms a antigen binding site. This is a hinge region which is rich in proline, and this is what we call as FC, fragment constant region. Okay, these are the different diagrams which have been shown in details. Now continuing further. Complementarity determining regions, CDR regions. Those are the regions where it binds with the antigen. So naturally, these CDR regions, where are they located? They must be located on variable regions of light chain as well as of heavy chain. And these CDR regions are also known as antigen binding sites, ABS, or fragment AB, like the other one which we will call it as a fragment FC, fragment constant, constant 1, constant 2, constant 3, heavy constant 1, heavy constant 2, heavy constant 3. But this, which is at the N terminal portion of light and heavy chain, together they form antigen binding sites, and these antigen binding sites are what? That is complementary determining regions. It determines the exact way how it will bind with the antigens. And as I have told you, they make up these antigen binding sites. They are made up of variable heavy and variable light. Okay. Now, little bit about hinge region. Hinge region, as I have told you, it is a flexible region rich in proline. Hinge region is found in IgG, IgA and D. What is IgG and D? That we'll discuss later. These are the different types of antibody. Basically, there are 5 types of antibody: G, A, M, D and E. So out of these 5 different types of antibody, hinge region is found in G, it is found in A and it is found in D. Me, that is ME, M and E, they do not have that hinge regions. Okay. And why I am telling you this? Because it will have implications in the next coming slides. Because these proline rich hinge regions, they are the target for proteolytic digestion. Proteolytic digestion means digestion with certain enzymes which will digest proteins. Which are those enzymes that we will study later? They are papain and pepsins. Okay. They are also rich in cysteine residue, that is disulfide bonds. As I have told you, IgM and IgE, they lack hinge region. And these IgM and IgE, they have extra CH4 domains. Normally, constant heavy 1, constant heavy 2, constant heavy 3. There are 3 domains, but one more section is there in case of M and E, and that is CD, that is CH4 domain. So this is something which is shown in. Okay. Now, Theselius and Cabot discovered the presence of antibody in serum, and that is why they called it is the immunoglobulin G, slowest moving fraction of globulin in serum, which contained maximum ability to impart immunity, which had the ability to react with the corresponding antigens. Now, the structure which we just now studied, that structure was basically discovered in between 1950 to 1960 by different scientists, and prominent among them were Rodney R. Porter and Gerald Edelman. In short, Edelman and Porter, they were the pioneers who studied the structure or who discovered the structure of antibody, as we had just studied now, made up of 4 peptides with variable and constant regions and presence of sugars, presence of disulfide linkage, as well as the presence of hinge regions. All these things and much more, they were basically studied in detail by Porter and Edelman between 1950 to 60, and for this they were awarded with Nobel Prize in the year 1972, because this was one of the basic structure that played an important role in this molecule. How did they discover this structure? They did it with treating these immunoglobulin molecules with the enzyme papain and pepsin. I will show you the diagram, this one. Let us assume that this is a complete molecule of immunoglobulin. When they carried out the digestion with enzyme pepsin digestion, then what happened? That what they obtained after each treatment with each enzyme, when they treated this immunoglobulin with a particular enzyme like either pepsin or papain, again they carried out electrophoresis, so that whatever the protein molecules that have been broken down will be shown in electrophoretic mobility in different components. So when they did this digestion with pepsin, they obtained two fragments. What were those two fragments? One was FAB2, AB2, fragment antibody. How many? Two fragments. And there was no fragment FC which was recovered. It was totally digested. So this was how it was. So when they digested with pepsin, what they obtained was FAB. Why it is known as? Because this is an antibody binding region. So we will call them as a fragment antibody. How many are there? One, and this is two. So we write it as a FAB2. Okay. Now why they obtained it like this? Because the mode of action was somewhere here. Pepsin acted here. So one fragment and these two fragments were obtained. And this one was completely digested. But say with the help of another enzyme, proteolytic enzyme, papain, the digestion was different. Why? Because when they digested with papain, they obtained three fragments. This was FAB, another FAB, and this FC region. But this FAB regions, they were not together, and therefore it was known as fragment antibody binding. This is separate and this is also separate, and fragment constant as one, because the mode of action of papain was here above this. So what will happen? This portion one, this is the second portion, and this one fragment crystallizable, FC. Why name C was given? C because it was able, one could crystallize that, fragment crystallized. So this was FC region. This was what they obtained it by digestion with papain. So found that it was because the mode of action was here. It digested this portion. Third, when they used mercaptoethanol, mercaptoethanol will destroy disulfide linkage. So this would be broken here, this would be broken here, and this will be also be broken. So what will happen? You get four different fragments and none of them is able to bind with the antigen. None of them, because for binding with antigen you require variable heavy as well as variable light. Both these things are essential here. Then and then it will bind, but individual peptide will not be. So when this particular mercaptoethanol was used for digestion, disulfide linkages were broken. It was reduction reaction. And therefore what they obtained was this, two heavy chains and two light chains. Now if when they combined all these together, then they formed or they devised or that gave an idea about the structure, how would be these four different peptides are linked with one another, and that ultimately gave you the structure of antibody. So this is the one. This is shown in diagram, the puzzle solved by Porter, Edelman, and even another scientist Nisonov. This is the cleavage by papain. When the papain, as you can see in red color, it is acting here. So what you have got, fragment antibody, two fragments, and this is fragment crystallizable. When proteolytic cleavage by pepsin is done, it attacks here, and what you have got is FAB2, and this is the one, FC region. But this is digested here, and therefore it is entirely digested. This is not at all in existence. Only two FAB2 regions.